plant tissue analysis, how to collect, process, and analyze the sample. We're going to talk about a lot of different things in this. All of these are very important, as collection of the proper specimen gives you a good result. Collection of a bad specimen gives you a bad result. In collection, we got to talk about soil sample. We need to know what is in the soil. We need to know what's available to the plant as far as nutrients go to find out whether or not the plant is functioning properly. We also have to take a tissue sample, because we need to know what's in the tissue. If it's not in the soil, it can't be in the tissue. If it is in the soil and is not in the tissue, then we got a different sort of problem we got to look at. We need to have a soil sample and a tissue sample if you want to do this right. A lot of these samples can be sent off to a lab for testing. Depending on what you're doing, there's going to be cost involved in it. You need to understand how these work. Then you need to compare the results you got out of the soil sample and out of the tissue sample to find out what sort of problem you're actually dealing with. When we do sampling, this shows you a field. What you do is you set the field off in a grid-shaped format. In here, we've got things collected at different spots within a grid. This allows you to pool a sample and then find out what's together. If you look at your field and you've got a very specific area that's different from everything else, you may want to sample that area separately. If you've got an area that's giving you a problem, and you collect sample from it, you mix it with everything else, it may come up to a normal level. If you take that area sample there and you send it in, you may find out that you've got a problem that you can deal with. In sampling, you want to take as many samples as actually possible. This kind of shows you about 10 rows by 10 rows and get about 10 samples out of it. We got one sample per row. The format you go in here really depends on the individual, what you're looking at. Number of sampling points is going to be determined by what you're looking for. Do you see the pattern in the field? If you've got one area of the field which is low and it's showing one symptom, one area of the field that's high that's showing a different symptom, you've got to take samples from each one of those areas. Sample the pattern. Always look at the pattern. Does the pattern follow drainage ditch? Does it follow differences in elevation? Is sample near a sprinkler or near a lake different from somewhere else? Always look at the pattern in the field. Be observant. Question always comes down to it. Do you pool the samples? If you've got a regular field and you don't have anything going on with it and everything looks about the same, go ahead and pool the samples. That way you're only going to run one sample. It's going to cost you a whole lot less. However, if you've got a high level and you've got a low level and you've got a depression, you've got a north to south slant and an east to west slant, you may have to get 4 or 5 different samples, especially if you got a drainage ditch running through the center of it. Therefore, you may not be able to pool the samples. Day of the week you do this on. If you're doing a regular soil sample, it's probably not going to make a whole lot of difference day of the week you do it on and send it in. You don't want to get a soil sample that's real wet. You want to have it basically dried down. If you're getting a tissue sample, never send it in on a Friday or Saturday or the day before a holiday, because you have to make sure that that sample is going to be received by the lab during a regular workday so that they can take that sample and possibly freeze it or put it in the refrigerator or start the drying process on it. If it sits over a weekend in a warm area, you're going to get mold growth on it. It's going to change everything that's going on. The day of the week is important to always make sure that your sample arrives next day, and that next day is a work day. Processing. We always say use paper bags. And the reason you use a paper bag is because a paper bag lets moisture out and it keeps humidity from forming on the inside of the bag. If you have a soil sample, you may want to spread it out to dry first. If you have a tissue sample and you're going to send it fresh, never use plastic because it'll allow mold to grow. Use paper. Don't put a whole lot in a paper bag. If you do put a whole lot in a paper bag, you may want to dry it out a little bit beforehand. They're going to process it for chemicals on the inside. You want to always avoid moisture. When they get the sample, they will dry the sample to a constant moisture. That constant moisture is very low. They want almost all of the moisture driven out of it. You continue to dry it until it doesn't change in weight. What that means is you've driven off all of the moisture. When we know that we don't have moisture in it, then we are able to say that of the dry matter, this is the material that you've got on the inside. When we dried it, normally we do 80 degrees centigrade for about 24 to 48 hours. You do these in paper bags. One thing, we used to have a forced air oven where we did it. If the forced air oven started to smoke, never open the doors. The temperature may get a little warm on the inside in there. It may allow for a little bit of charring, but if you open those doors, what will happen is you will allow oxygen to come in. As soon as you do that, you will have combustion. I had a student once who lost 6 months' work because it started to smoke a little bit. She opened the doors, they all burst into flame. Once it's dried down, you're going to grind it to a powder. Then we can analyze the powder. We do this with what we call a mill. We use a Wiley mill. A Wiley mill is a rather strong sort of mill that chops it into very fine pieces. It's got a screen in it to make sure that what we're getting is almost a dust-like material. That way we can break it down and analyze the dust-like material. Some people use a mortar and pestle. You can do this with liquid nitrogen. You freeze the material. Once it's frozen like that, it cracks very easily. You can then analyze that material. You can use something like a blender. They sell little coffee grinders and you can use those as well. You put your sample in there, you zap it through and you get it to a very small, fine texture. Then you can go and analyze the sample. We do it in a process we call ashing. Ashing is going to leave only the mineral part behind. All of the carbon is going to be converted into carbon dioxide. All of the hydrogen and oxygen is going to be converted into water or will be evaporated out. You're going to have the mineral part in the periodic table come down to the form of the metals. The non-metals tend to be oxides and they tend to volatilize off, which means there are special manners by which we analyze them. Carbon dioxide is released, as are oxides of sulfur, nitrogen, and other non-metals. There are two basic formats for ashing. You can do a dry ash, which means you incinerate it at a high temperature. This is often around 1,000 degrees. It causes the carbon to combine with oxygen to give you carbon dioxide. There is a wet method where you mix it with concentrated acid, either nitric or perchloric, or a nitric perchloric base. You can use a sulfuric acid. It will form soluble salts. These can be used. When you do a wet method, basically you're going to heat it and boil it for a period of time. We have what we call a Kjeldahl method, which is done with nitrogen, and we'll explain that later. This has a reagent added to it that's going to trap the nitrogen that is released. We can sample the nitrogen. We can sample it in a couple of different forms. Today, one of the major ways we do things is through ionization. In ionization, you can actually take a dry sample, put it into an ionization chamber. It will be bombarded by particles. The particles will cause all of the nutrients to be released. You can tell exactly what's on the inside of it. Wet ashing. We start out in a fume hood that's going to use acid. It is going to generate some really caustic vapors. We want to be very careful about this. We have a heating block. The heating block is going to heat these to a boiling point. You're going to have the tubes hold the sample on the inside. It's going to boil in that acid. It is going to only retain the mineral portion. You can see here there's a tube that we use. It's got a little line on it. This is about 1/3 of the way up. This is the line where you would add your acid to it. The sample is normally added as a small dry powder. Then you would put it in and it basically would boil. This is what they look like in this specific holder for these particular types of tubes. You place the holder onto the heating pad. You can see the tubes drop down in height. That's because the other part is inside of the heating element, which means it's getting very hot. It's maintaining it at a constant temperature. If you lift up this little grate on the inside, you can actually see where the tubes are. The tubes extend down into the holes in the heating pad on the basin. That is going to allow for boiling of the material. This is a wet ashing. Dry ashing uses an increased temperature to oxidize everything. We take it, we put it into an oven. This oven will reach about 1,000 degrees. You can melt gold in here. It gets to be very hot. We place the sample inside of little crucibles. These will get red hot and it will cause everything to vaporize with the exception of the metals. What you've got left over can be analyzed. You let it cool down and then you can add liquid to it to absorb the material. And then you take that, you place that into the analysis machine. There are a couple of different methods of doing this. We have a colorimeter where you add it and it gives you a stable color that you can absorb. We've got what we call an atomic absorption spectrophotometer, which will tell you specific materials. I remember doing this with calcium, potassium, and magnesium. It works very well with ions. There is a procedure that you would use. Then there's titration. In titration, basically you add an indicator. You have a material of known concentration that you use for the titration part. As it does this, it's going to provide for a color change, which is going to be visible